Welcome back to the next step here. So what we're going to do is look at meiosis in this case. And meiosis is a specific type of cell division that is done only in the gonads, and it is the making of sex cells. Sex cells are often called gametes, right? And I've got a nice drawing for you here to visualize. Some of these gametes are sperm cells, right? And some of them are egg cells. So the sperm cells are super tiny, the egg cells are much bigger, right? And what will happen eventually is they will join together in the process of fertilization, and that will yield a single zygote. As I have referred to it, the zygote is that single cell that will begin to divide mitotically, generating identical daughter cells, and with a few millions and millions and millions of divisions, and with one complete genome, this will eventually make an adult. Da-da-da-da, with at least 18 years, I suppose, of dividing and dividing and dividing and dividing. So in the process of doing this, we don't make identical cells like the parent cells because there would be too much DNA in the zygote. So as a result, we have to half or cut in half each of the parent's DNA and then put that in the gametes. So to do that, we do the following steps right here. Number one, the DNA gets replicated just like it does in mitosis. The difference is that instead of just skipping to the sister ripping part, we go to separating the homologues, and that means each parent will give one of each homologue exactly. Okay, and then we will separate the sisters' chromatids that are identical after the homologues have been separated. So it's a little more complicated than mitosis. But let's take a look. In this particular case, we've got a simple organism here, right? And then I've drawn it here with two homologous pairs like this. And the big difference here is that, in fact, in metaphase 1, which is what this is, metaphase 1, they line up with their homologues. And that's super important because that means these spindle fibers can attach like this, and then, of course, this is all meant to be inside a cell, right? So I can make a cell membrane on the outside. But that means that now there will be separating the homologues. So when you're making sperm cells or egg cells, the first step that happens is they line up with their homologue and they get separated. Now, there's also, I've drawn some little arrows in here that we'll take a look at those in a minute. So when I come back and talk about what's happening there. The idea that I've drawn stripy on the right side over here, and I've got the other, and then I've got some clear ones on the right side as well. And these are going to be separated, like the pairs themselves have this random, they kind of dance around in a circle here, so they go like this, and they spin around, and they take turns being on the different sides, and when the spindle fiber connects, bing, right here and right here, it could be on either one of the pairs, either the clear one or the stripy one. And as a result, each of these descendant cells that come out of this will have a combination of stripy and clear chromosomes. So let's go on to the second step then. So after they've separated, they would look something like this down here. So I would have two cells, and those cells, one with each homologue is in there, right? There's one homologue, there's the other homologous pair. They're paired with these guys, but they're no longer in the same cell, right? They're in different cells. And so the next thing that happens is the sisters get pulled apart. So I can put spindle fibers on here like this, and these spindle fibers indicate that these little cables grab each of the sister chromatids, which are nearly identical at this time, and then they separate them into single cells that are haploid cells, which means they only have one of each kind of chromosome. They're not diploid, which would be two homologues of each chromosome. And then each of those, and to do easy visualization here, I've used the sperm cells, so you can say, put these tails on here, and now you know those are gametes. okay? They are each unique in this case because the way I've explained it is you might think that the two on the left here are more similar and that the two on the right are more similar, but some things have happened along the way to make that not the case. So let's go take a look to see what that is. I might not have mentioned that the second lineup on the centromeres is called Meiosis 2. Okay, so notice that you've got centromeres in here in the middle. Two lineups, right? First lineup with the homologues. Second lineup is with the sisters on the centromere, all right? In the beginning, there's a step right before this one when these chromosomes are kind of condensing and getting ready for division after they have replicated the DNA, before they're lined up with their homologue, they go through a thing called prophase, where they make homologous pairs. So the homologues find each other, but they're not lined up yet. And so this is kind of a cool time when they're paired right here. I have them paired up, and you see the two together, but they're not lined up yet with their pairs. And what happens in this case is the DNA is so similar because the genes are the same, but sometimes the alleles are different, but not even always, but sometimes. And so there's the thing that's called crossing over. And I'm going to write it out neatly for you right where it had it before, which is called crossing over. And that means that the two homologues swap chunks of DNA. And what I mean by swapping chunks of DNA is that quite literally, when you put two long strands of DNA together that are virtually identical and they're piled next to each other, they break sometimes and then they can get reattached on the other side. So for example, if this left-hand chromosome is the blue chromosome like this, and then I have a red chromosome over here, and they break at this point, when they get reattached, the blue one ends up being on this side with the red rest of the chromosome, and the red one ends up being on this side, right, with the blue rest of the chromosome. As a result, this is called recombination, and it means allele combinations are recombined, right? Because now you've got this blue base with the red top, and you got this red base with the blue top. So you're recombining alleles like eye color and thumb shape and hair color. They are in new combinations. Okay, so that gives you new combinations, and as a result, when we get, here's another drawing of how that works. In fact, they happen many times, so lots of reshuffling occurs in every homologous pair. So at the end of the game, you've actually shuffled up a whole bunch of your genome, and if I wanted to make this blue and blue, right, then I would have red tips on here, and maybe this red blue section would go over here. It gets pretty wild and crazy, I guess. You could have some red over here. This one might be red. This might be blue on this side down here, and this would be blue. So basically it becomes red chromosome. So this all becomes one big mishmashed up collection of genes. So that crossing over is important because the crossing over is a reshuffling, right? But crossing over also causes genetic variation. And what that means is the offspring will have combinations of the parents' genes that the parents didn't have. Wow. Okay, so that happens. And then we also recall that this section here, where these two were dancing, so to speak, in their little dancing mode and spinning around, and we know that we had stripy on the right and we have clear on the right for this other chromosome. This process is called independent assortment, which is the process by which which homologue ends up in which daughter cell is independent for every pair. So that means that basically when you get to the end of the game, I can tell you that every sperm is unique and only one can fertilize an egg. Everyone's unique because of crossing over and independent assortment. And so that's very exciting. So that is the story of meiosis.